This is HAP 201, Week 2, Chapter 29. We'll begin with a very short and superficial overview of development of the fetus, as well as some changes in the mother during pregnancy. We'll be looking at some key milestones of development, some key indicators of pregnancy, and take it from there. Developmental biology looks at the sequence of events that occur from the moment of fertilization, when the nuclei of a sperm and an ova unite, to the birth of the infant, and then development into an adult organism. The embryonic period of gestation is from fertilization through the first 8 weeks of development. It's a very sensitive time. For the first few weeks, the mother may not even realize that she is expecting, and it's a time when the developing baby is very sensitive to external and internal factors that can lead to serious defects in development. So even though the mother may not be aware, it's important that they maintain health if there's any chance at all that they could become pregnant. The next remaining weeks of the pregnancy are called the fetal period, beginning at week 9 and continues to week 40. And the neonatal period covers the first 28 days after birth, so up to 1 month postpartum. Fertilization, again, usually occurs in the uterine tube of the female. And note that sperm that are ejaculated are not capable yet of fertilizing the secondary oocyte. These sperm have to undergo further changes in a process called capacitation, and these allow the sperm to penetrate the secondary oocyte and fuse membranes with that of the oocyte. Capacitation involves exposure to substances released in the female tract, especially those in the fallopian tubes. So as several sperm reach the secondary oocyte, they are attracted to it and begin to release their acromosomal enzymes, breaking down the layers around the secondary oocyte, and one for the sperm that can reach the plasma membrane of the secondary oocyte after moving through the corona radiata and the zona pellucida. That is the sperm that will fuse with the secondary oocyte, trigger the completion of meiosis II in that cell, and their genetic information will combine to form the zygote. So fertilization again involves first the breakdown of the protective layers around the secondary oocyte. The corona radiata is comprised of granulosa cells from the mature follicle with which the secondary oocyte was ovulated, and between those cells and the membrane of the secondary oocyte is a clear layer of glycoprotein called the zona pellucida. So acrosomal enzymes plus the strong swimming action of the sperm help to break down those two layers, and the sperm that's able to clear a path to the plasma membrane of the secondary oocyte will be the one to fertilize that cell. And by fertilizing it, it initiates the completion of meiosis II in the secondary oocyte so that it becomes an ovum. Another polar body is produced, which degenerates off to the side, and then the genetic information from the two gametes are combined. So fertilization involves the combination of the two nuclei, or because they're in the gametes, they're called pronuclei, and a single cell, first diploid cell, created by that joining of nucleic acids, is called the zygote, and is the first cell of the embryonic phase. Again, this usually takes place in the uterine tubes, and After about 24 hours, there's that first cell division. Following that, the cells divide quite rapidly to form the next stages of the developing zygote. Okay, as we mentioned in the week 1 slides, the growing ball of cells take up to a week, 4 to 5 days, before they implant in the uterine cavity. So after several cell divisions, the zygote develops into a solid ball of cells called a blastocyst, floats into the uterine cavity, bounces around until it finds an appropriate site within the endometrial wall for implantation. Note that an implantation usually occurs within the body of the uterus, not the fundus, not the cervix. Those are not consistent with a healthy pregnancy and will lead to a malplaced placenta. So the most common and most beneficial region of the uterus in which the blastocyst should implant is the body of the uterus. So that process is complete at about 1 week after fertilization, and if you recall, fertilization is going to happen around the time of ovulation. So this is about 21 days in the female cycle. This is normally when the corpus luteum should begin to degenerate. Because of this implantation, further development, you can see in the last image of this diagram that the blastocyst is now having cells separate and begin their differentiation. The trophoblast will contribute to the placenta, and that region of this developing embryo secretes the hormone HCG, which feeds back to the ovaries of the female to promote the secretion of hormones by the corpus luteum, estrogen and progesterone, and those will support the pregnancy itself within the uterus. So here are the events of the first week post-fertilization, and you can see the developing zygote embedding and developing within the endometrium of the uterine wall. Great diagram showing you the progression of events. This early stage of development involves many cell divisions. It's quite rapid, and these are the blast cells. These are the stem cells that will give rise to all the structures of the embryo and the supporting structures. So the embryo develops an embryonic disc and 4 extra embryonic membranes. The embryonic disc will divide into the structures that will make up the fetus and includes 3 distinct tissue types, while the extra embryonic membranes will form the supportive structures such as the amniotic sac, the umbilical cord, the placenta. Now, because the cell division is so rapid at this time, the basic cell types are being laid out. Any errors in DNA copying and any mutations that occur can lead to serious developmental defects. So this is a stage where the embryo is very susceptible to factors that disrupt normal development. Those are teratogens. Great word to say, teratogens. And there's a long list of teratogens. If you have time, just Google teratogens. Everything from alcohol, cigarette smoke, certain vaccinations, certain viruses can act as teratogens. Several chemicals can have deleterious effects on the developing embryo as well. So it's very important that a healthy lifestyle is maintained during this time. Here's a diagram just showing you the structures of the embryonic period. That little alien in the middle is present after only a few weeks of gestation. So it doesn't take long for the cells to begin to differentiate and segregate to form the structures, the primitive structures of the various organs and systems of the developing infant. So here we have a diagram depicting the early differentiation of the cells within the embryo into three distinct tissue types, or three primary germ layers arising out of the embryonic disc. There is the ectoderm, mesoderm, and endoderm, and these are surrounded by the supporting structures, the extra embryonic tissue. So the ectoderm will differentiate into tissues that make up the epidermis of the skin. And interestingly, even though that is a very simple type of tissue, the same germ layer will give rise to the highly specialized tissues of the brain and other structures of the nervous system. The middle mesoderm is a loosely organized connective tissue called mesenchyme. You may have heard that word before, or mesenchyme. And it will mature to form connective tissues and muscle tissue. So blood, muscles, bones, other connective tissue types. The inner endoderm will give rise to the epithelial tissue that lines all the tubes and ducts of the body, including the endothelial lining of the blood vessel. So these are the three germ layers that give rise to all the different tissues that will make up the organs of the developed fetus. Now please pay attention to this slide. The placenta is a specialized organ that develops during a pregnancy. It's formed in part by cells of the embryo and cells of the endometrium of the uterus. So specifically, the chorionic villi of the embryo and the decidua basalis of the endometrium give rise to this specialized organ. The placenta has several roles. Number one, it is a site of nutrient and waste exchange across it by diffusion. But note that in a healthy pregnancy, the placenta does not allow mixing of blood, but substances from the blood of the fetus can be exchanged with those from the blood of the mother selectively. So the placenta is fully formed and carries out all its functions by the end of the third month. And the placenta then takes over hormone secretion from the corpus luteum, which was maintaining estrogen and progesterone secretion under the influence of HCG from the primitive or still developing placenta. The placenta also forms a selective barrier, as we said, that allows for nutrient exchange and gas exchange. Also note that maternal antibodies, specifically class G, can cross the placental barrier and enter the circulation of the fetus. This will confer some passive natural immunity on the fetus. So the infant is born with antibodies preformed by their mother. These do not last forever. It's just short-term temporary immunity. Wastes are removed from the embryonic blood across the placenta. They enter the mother's bloodstream and then are excreted from the mother. Carbon dioxide by exhalation, most other wastes through the urinary system. We mentioned that the placenta takes over hormone production, not only of estrogen and progesterone, but also produces several other hormones that are specific to the pregnancy and support not only the developing fetus, but also the mother, and cause several changes in the mother. The major one being further development of the mammary glands, preparing them for lactation. Note that the teratogens we mentioned on a few slides ago can cross the placental barrier and therefore cause serious developmental abnormalities and sometimes death, especially in the early stages of a pregnancy. Unfortunately, many pregnancies that do not go full term end in the first 8 weeks of the pregnancy, when that developing baby is very susceptible to these harmful substances. In terms of developmental defects, they often occur in the heart and the brain or other structures of the nervous system. If the fetus has experienced an error in development that is incompatible with life, that pregnancy may self-terminate. All right, we're going to quickly move through the stage of the fetal period and just note some key milestones of development. So after the first 8 weeks, which is the embryonic period, the developing child will then enter the fetal period, which covers weeks 9 through 40. During the fetal period, tissues and organs that develop during the embryonic period continue to grow and differentiate. So please note that by the 8th week of gestation, all of the major organ systems are already present in their rudimentary form. We're talking about 2 months into the pregnancy, and the heart is already pumping, the nervous tube or the neural tube is already there, limbs are in place, the precursors to the eyes, internal organs, they're already present in their rudimentary form. As you can see from the diagram on the left, E doesn't look very human, but it looks like something. And by 13 weeks, the fetus definitely takes on a more human appearance. The body is a little more proportional to the head. The limbs elongate. You see the structures on the head, such as the ear, the eyes, the nose, the mouth, etc. Okay, and that's only 13 weeks gestation, early in the pregnancy. So again, during the fetal period, very few new structures appear, and at this time, there's a better chance for survival of the pregnancy. The fetus is less vulnerable to damaging teratogenic effects. However, minimal exposure is the recommendation to maximize the health of that fetus. During this time, there's rapid growth, enlargement, and further maturation of the organs, especially towards the end of the pregnancy, where the infant pretty well doubles in its weight. By the 34th week, towards the end of the pregnancy, the fetus has all the organs in place. The infant will assume an upside down position in preparation for labor and delivery. The lungs are almost completely mature and producing enough surfactant to support normal respirations and opening of the lungs on their own. So again, gestation is a time of prenatal development. It encompasses 40 weeks divided into three 3-month trimesters, so first, second, and third trimester. So we already mentioned that the first 8 weeks occur in the first trimester. That's the embryonic development and early fetal development, where all the organ systems are laid down. During the second trimester, next 3 months, there's further development of those organ systems, and the third trimester is really about putting on weight and further development of the organs and getting them ready for extrauterine life. So again, by the 8th week of embryonic development, all the organ systems are present in their rudimentary form. All right, some key milestones of development. As I mentioned, already early in the development, by 3 weeks, the notochord, which will be the basis of the central nervous system, is already present, and the heart is already contracting as a single tube. 3 weeks. At 6 weeks, structures of the respiratory system begin to develop, and circulation as well. And cells from the amniotic supporting structures will give rise to the blood cells. At 8 weeks, again, everything's already present in basic form. The head represents about 50% of the total body length, but all major brain regions are present, and the skeleton of the fetus that began as cartilage begins to ossify into bone tissue. All right, 9 to 12 weeks, this is when the mother is usually going for her first ultrasound and her first Doppler test to hear the heartbeat and to check the assessment of the infant. The bone marrow is making its own blood cells, and the genitals of the infant may become visible. The external genitalia are present, and visible if the child decides to assume a position in the uterus that can be detected by ultrasound. All right, moving into the second trimester, week 13 and beyond. The eyes are blinking and sucking motions are occurring in the fetus. The body begins to outgrow the head, so the body parts become a little more proportional. The bones are distinct with joint cavities, and the eyes and ears appear to be moving towards their normal position. It's just about the morphological changes. And by 16 weeks, infant looks human. 16, 17 weeks, getting to the 5th month, this is when the mother might experience quickening, which are the sensations associated with the movement of the infant. So the baby starts jumping. The expression is often used. The fetus becomes covered with soft, downy hair, and vernix caseosa covers the skin as well. It's like a cottage cheese-like substance. It protects the infant and helps to incubate it within the amniotic sac. Halfway through the pregnancy, in the middle of the 2nd trimester, the fetus is experiencing substantial weight gain, and even if they are delivered prematurely during this time, they have a very good chance of survival with assistance after 27 weeks gestation. So that would require intensive care, definitely ventilatory support. But the chances to survive and live a healthy life a few years later, very good. Fingernails and toenails are visible. And as we mentioned, in the male reproductive system, the testes that develop in the pelvic cavity begin to descend down into the scrotum at about the 7th month. In the last trimester, the last few weeks, the fetus essentially is putting on weight and becoming more proportional with respect to the length of the body comparable to the head. Surfactant production is adequate by about 32 weeks gestation. So again, the infant has a very good chance of survival and even breathing on their own, especially if they're born after 35 weeks gestation. So here's an overview of the developmental stages, noting the weight and the length of the infant. Please feel free to open this interactions animations and track the development. Now for the female, there are several indicators of pregnancy. So please recall that ovulation does not come with a distinct marker. No one wakes up in the morning and says this is ovulation day. But there are indicators that fertilization, implantation, and pregnancy have occurred, and as the pregnancy progresses, the signs go from possible and probable to very likely and positive. So early changes in the mother include changes in the breast. They become a little more sensitive, may become bigger. Strong indicator of pregnancy is the absence of menstrual period. So fertilization would occur around day 14. 2 weeks later, the embryo should implant and begin secreting HCG so that estrogen and progesterone levels remain high. The endometrium is not slought off, and after 2 weeks, the woman would be expecting but would miss her period, and that definitely will continue for the next few cycles because it has been interrupted by the pregnancy. But note that the absence of a period, although a very strong sign of pregnancy, is not a positive sign because there can be other causes for amenorrhea. Morning sickness, which includes nausea, vomiting, and lack of appetite, can occur anytime in the pregnancy. It really varies depending on the woman and the particular pregnancy, but they may start to occur early in the pregnancy, anywhere from 4 to 3 weeks onward. For some women, it subsides by the end of the 1st trimester. Others experience it throughout the entire pregnancy. Urinary frequency occurs 6 to 12 weeks into the pregnancy. Not only is a growing uterus compressing or pushing down on the uterus, but her blood volume has increased significantly, and there's extra wastes to clear because she's also considering or also compensating for the metabolism and development of the embryo slash fetus. So urinary frequency occurs. Fatigue is a very strong indicator, and we said that towards the second trimester, she'd feel movements in the abdominal region. The contractions that occur towards the end of pregnancy are called Braxton Hicks contractions. They are not labor contractions, but are often confused with them. And a pregnancy test, a test for HCG, will probably show up positive. And the ones these days are pretty sensitive. They can probably detect the HCG levels in the urine closer to 3 or 2 weeks of pregnancy. Blood tests even earlier. But please know that these are all possible and probable signs. All of these, the changes in the breast, the urinary frequency, fatigue, etc., even the positive pregnancy test, could be due to other causes. So they are not definitive indicators of pregnancy. However, positive signs include the visualization of the baby on an ultrasound. Can't deny that it's there. Auscultation of fetal heart sounds, which can be detected as early as 6 weeks, especially with the Doppler apparatus. And when the practitioner can palpate and detect fetal movements, that is a positive sign of pregnancy. In a full-term pregnancy, the uterus will grow to nearly fill the entire abdominal cavity. In doing so, it will displace the other abdominal pelvic organs, such as the intestine, the bladder definitely will be affected. Weight gain occurs due to the growth of the fetus and supporting structures, including the amniotic fluid. But note that a female should not expect to put on excessive amounts of weight during a pregnancy. So if a female went into her pregnancy at a healthy BMI, a healthy weight, she can expect to put on maybe 25 pounds total for the entire pregnancy. And only 7 of that is baby. The rest of it is supporting structures and excess fluid. And they will be lost pretty soon after delivery, and then it takes up to a year to regain pre-pregnancy status. So again, the adage that a female is eating for 2 when she's pregnant is not true. At the most, you're eating for 1.4, and the caloric intake can increase about 500 calories a day, but excessive amounts of food do not have to be consumed during a pregnancy, because the weight that is gained, that is excessive, is very hard to lose after delivery. The breasts become larger, the ligaments throughout the body begin to relax, and because of the growing uterus, the female center of gravity is shifted, and to maintain her balance, she often develops temporary lordosis with the curved, exaggerated lumbar curve. You may have seen women walking with their hands behind their hips, trying to support their back. That will correct itself after delivery. So note in this diagram that as gestation progresses, the fundus of the uterus will rise in the abdominal cavity. At about 9 months, the infant is pretty well sitting under the ribs. It can make it very difficult to breathe. The vital capacity can be decreased significantly, but everyone will be okay in a few weeks. Also, by this time, the fetus should assume the birth position, head down, pushing down on the cervix, and towards the time of delivery, the cervix will thin. That's effacement. It will also dilate, so it will separate and open up, allowing for passage of the infant. The largest part that has to be delivered is the head. So head first deliveries are the safest and the easiest in terms of vaginal deliveries. Once the head is out, everything else just pops right out. All right, please note this. There are Significant changes in the cardiovascular system of the mother. Note that there's a significant increase in their circulating blood volume up to 40%, as well as their red cell mass due to the increased metabolic demands placed on the mother by the fetus. So even though their blood volume goes up significantly, their cardiac output also goes up, but their blood pressure should not. In a healthy pregnancy, the blood pressure should not go up. It should stay the same and sometimes actually drops during the second trimester. So a rise in blood pressure is a pregnancy complication. If it's severe, it becomes preeclampsia, and that can then lead to seizures and coma for the mother in a condition called eclampsia, which definitely should be avoided and should not be occurring. So what prevents the blood pressure from rising even though the blood volume has increased significantly? There's a corresponding compensating decrease in the vascular tone throughout the body. So systemic vasodilation will lower the pressure even though the blood volume is increasing the pressure. And by the third trimester, the mother should be back to her first trimester blood pressure levels. Blood pressure, blood sugar levels, and the presence of amino acids and proteins in the urine are things that are assessed for with every visit during the pregnancy. Sugar in the urine indicates gestational diabetes, and proteins in the urine are indicators of pregnancy-induced hypertension and preeclampsia. The digestive system and appetite centers might be affected. So for some women, that's an increase in appetite. In others, it's a decrease in appetite. We talked about morning sickness, which occurs in response to the rising hormone levels. That can be corrected or managed by eating smaller meals more frequently and switching to a lower fat diet. So dry foods, high in fiber, smaller meals often help alleviate the morning sickness. If it's very severe and the female is vomiting excessively and losing a lot of fluids, electrolytes, we call that hyperemesis gravidarum, and can lead to dehydration and malnutrition. Under those situations, the mother might be assigned to bed rest and IV treatment. Heartburn is not uncommon as the fetus rises up in the abdomen and the intestine is displaced. Also note that the increase in the intra-abdominal pressure decreases the competency of the lower esophageal sphincter. That's the junction between the esophagus and the stomach. So heartburn and reflux of acid in the esophagus is not uncommon during pregnancy. At the other end of the tract, the mother may experience constipation, so increased water intake will not only help to alleviate that, but also help with the morning sickness. The urinary system increases its activity, increased urine output to, again, clear the excess wastes. Also note the increased blood volume that has to be managed. And we talked about the growing uterus putting pressure on the bladder, increasing urinary frequency and urgency. And it can also cause stress incontinence where the external sphincter is no longer as competent as it was pre-pregnancy, and then anything that increases the intra-abdominal pressure can lead to small leaks of urine between conscious urination or micturition. So laughing, coughing, sneezing, those kind of things can trigger small releases of urine. In terms of respiratory function, the respiratory rate can increase significantly to supply additional oxygen to both the increased metabolically active tissues of the mother and the fetus. In the skin, again, this varies very much from woman to woman, pregnancy to pregnancy, but hormonal changes can cause stretching in the skin, and of course in the abdominal area and the thigh sometimes if there is weight gain, fluid retention. The stretching of the skin can cause tears in the underlying dermis, leading to stretch marks. Stretch marks never ever go away. They may decrease in appearance, but they cannot be removed, unfortunately. There's increased subdermal fat, which also contributes to stretch marks. Some females experience hyperpigmentation due to increased melanotropin, and it creates a darkening of the face called cloasma, or the mask of pregnancy. Some females have a hyperpigmentation of the line running through the rectus abdominal muscles, and it's called a linea nigra. Some women experience increased growth in their hair and nails. For others, their hair breaks or becomes less manageable. Again, it's very different how each female reacts to the hormones of pregnancy. Sweating is not uncommon. Neither is pruritus, which is itching, dry skin, itching, stretching of the skin. The hormones may also be contributing to the. We will now consider some of the basic principles of inheritance that occur when the genetic information from the parents is combined to create the genetic information of the newly developing individual. A lot of this should be reviewed for you, but again, please read through the chapter and the sections that you are unfamiliar with, and also go back to chapter 3 and look at the processes of cell division, especially meiosis, if any of this is unclear to you. The production of gametes we've looked at. It's quite complex, but also very well designed in terms of the separation of the chromosomes into haploid cells, so that half the chromosomes from the mother and half the chromosomes from the father can then unite to create a unique combination of genetic information that will define the characteristics of the new individual. So the genes encoded in the DNA of these cells will pass on hereditary traits from one generation to the next. They also direct the life functions of the individual. So it's a necessity that no species can live without. The branch of biology that deals with inheritance is called genetics. So even though a child might have certain characteristics or features that resemble one or the other parent, each individual is unique, and that is due to the separation of the chromosomes into the gametes, and then the arrangement of those chromosomes, which ones segregate into which gamete, how they express themselves when those chromosomes are combined in the zygote, and there are other sources of variability. So just random segregation of the chromosomes into the gametes introduces variability. That's why siblings may look like each other, but they're still very unique. Also, variability is introduced by exchange of genetic information. Physically, chromosomal pieces can be exchanged during meiosis 1 so that the chromosomes in the gametes do not resemble the original chromosomes in the progenitor cell. Genetic information from the maternal copy of a chromosomal pair can be exchanged with that from the paternal chromosome in a pair. Also, there's random alignment and segregation of the chromosomes into the daughter cells. So each gamete has a very unique assortment of chromosomes depending on how the chromosomes aligned in their pairs during meiosis 1. So when the chromosomes from the sperm combine with those from the mother in the oocyte or the ovum, they restore the full complement of chromosomes, so 23 new pairs are created, and 1 chromosome in each pair came from either of the parents. So these are called homologous chromosomes, and there are 22 pairs of chromosomes that have the same length and the same number of genes encoded in each chromosome. 1 pair is unique, those are the sex chromosomes, and 1 of the pair came from the mother, it's always an X chromosome, and the other chromosome in the pair came from the father. If the father's gamete had an X copy of the chromosome, then the zygote develops into a female because it has an X from the mother and X from the father. If the sperm cell had a Y chromosome, and that's what the father contributes to the zygote, that fetus usually develops into a male because it has 1 copy of the X from the mother and the Y chromosome from the father. The Y chromosome is much shorter than the X chromosome, so there are several genes on the X chromosome that will not be matched by a pair on the Y chromosome, meaning that the form of that gene carried on the X chromosome will determine the expression of that trait. We'll look more at X-linked conditions towards the end of this section. So each of the 2 homologous chromosomes contain genes that control the same trait, or together their combined expression determine a given trait. Alternative forms of a gene that code for the same trait and are therefore located at the same location or loci on a homologous chromosome pair are called alleles. So an allele is an alternative form of the same gene. Earlier we used the example of eye color. Eye color would be the trait, and there's different forms of the eye color. Blue versus brown or green, or variations of those colors. Mutations are permanent changes in the gene sequence that can lead to a different variant. Mutations occur pretty regularly. In healthy cells, they are detected and corrected and not passed on, but sometimes they are. Sometimes they confer new wonderful traits or forms of a trait. Often they can be harmful or deleterious. Alright, so back to the homologous pairs. When the forms of a gene, or the alleles, create the same form of the trait, they're both the same, we say that the individual is homologous for that trait. So back to the eye color. If the eye color site on the male chromosome of the pair is brown, and it's also brown on the maternal chromosome, then we say that that individual is homozygous for that eye color. If the alleles encode for different forms of that trait, then the individual is genetically heterozygous for that trait. We represent the various forms of the trait with letters. So capital letter for the dominant form of the trait, lowercase letter for the recessive or alternative form of that trait, and on the next slide we'll explain what dominant and recessive mean. The dominant allele is the form of that trait that when present is a form that is expressed. It can be present on one of the chromosomes or both of the chromosomes. If it's present on either, then that is a form of the trait that becomes visible. And we denote that by a capital letter. For example, broad lips are dominant over thin lips, so we'd represent the dominant or broad lip form with a capital B, and thin lips would be denoted by lowercase b. The trait, or the form of the trait rather, that is masked by the presence of the dominant, or the form of the trait that has to be present on both copies of the gene in order to be expressed, is said to be the recessive allele. So if it is present on both copies, that form of the trait will be expressed. But if it is present on one and the dominant form is present on the other, its expression will not be visible. The dominant form of the trait will be visible. So when we put this all together, we have to consider two features of inheritance: genotype and phenotype. Genotype refers to the combination of alleles that are present in a gene pair. So is the individual homozygous dominant for a particular trait? Are they homozygous recessive, so two copies of the recessive form of the trait? Or are they heterozygous for that trait, two different forms or alleles for that trait? That's a genotype. What it looks like, or what that trait looks like, how it is expressed, refers to the phenotype. So what is the overall outcome of that genotype combination? That is the phenotype. So the dominant phenotype is expressed when either one or two copies of that dominant allele are present, so the homozygous dominant or heterozygous form. The recessive phenotype is only expressed when the individual is homozygous for that recessive trait. We can denote the genotype for a particular trait, again, with upper and lowercase letters, and you can do that for the mother and the father when considering what possible combinations of alleles may be present for a given trait in the offspring. And when we do that, we arrange them around a set of quadrants called a Punnett square. So in this diagram, the mother's potential alleles are denoted in the red circles and the father's in the blue sperm cells. And it's possible that one of the gametes produced from an original eugenic cell could have one form of the allele and the other will have the other form of the allele. Same thing with the sperm. So because of random assortment and random fertilization, there are 4 possible combinations for this particular trait. And in this case, we're considering a metabolic disorder called phenylketonuria, which is the inability to metabolize one of the amino acids, phenylalanine. So the healthy state is denoted by the uppercase P, because the disease state in this case is the recessive trait. So this mother is heterozygous for the healthy gene and the unhealthy gene, as is the father. Kind of hard to see with the upper and lowercase p's. Now, the possible combinations of the alleles that are formed in the gametes will result in a 1 in 4 chance that a pregnancy will result in a homozygous dominant individual for that trait, which is indicated by the 2 uppercase P's in the upper left quadrant. There's the possibility that the combination of the gametes from the mother and the father will create a heterozygous individual. So 2 out of 4 chances that that could happen, or 50% chance, and a 1 in 4 chance that with each pregnancy the infant may inherit a defective P gene from the mother and the defective P gene from the father, resulting in the homozygous recessive individual that does develop this condition. This table indicates selected hereditary traits. Note that some abnormal or disease conditions, such as diabetes insipidus and Huntington's disease, are actually dominant traits. Huntington's is unique because the disease itself, although it's genetically present, it doesn't manifest itself until later in life, after reproduction. So an individual may be heterozygous or dominant for Huntington's, but the condition itself will not manifest until later in life. So the point here is that dominant does not always correspond with healthy. Sometimes the dominant trait is the abnormal or disease trait. So what we've described are your classic inheritance patterns, but note that there are some exceptions to this rule. So alleles that code for some traits might come in 3 different forms, or the dominant may be the disease, as we just saw. Also, there may not be a dominant versus a recessive. They may be equally expressed, or there may be an intermediate phenotype in the heterozygous individual. So on this slide, it just describes some of the errors that can occur during cell division in terms of chromosome segregation, leading to excess or deficient numbers of chromosomes in the cell. You may be familiar with Down syndrome, which is genetically called trisomy 21. There was uneven separating of the chromosomes, and one of the gametes actually included 2 copies of chromosome 21 and add the other copy from the other parent. That leads to 3 copies of one chromosome, and it creates very distinct characteristics in the offspring. So far we've been considering simple dominant recessive patterns of inheritance, where in the heterozygous genotype, the dominant phenotype will be expressed, which is your classic simple dominant recessive pattern. Let's consider a variation of that, which is incomplete dominance, where in a pair of alleles or different forms of a trait, neither form is dominant over the other. So of course in the homozygous individuals, they'll express the trait associated with that allele. The intermediate phenotype, however, is expressed in the heterozygous individual. So the person that has 2 different copies of the allele, or 2 different versions of the allele, will have an intermediate phenotype where one is not dominant over the other. So before I get to the physiological example of sickle cell disease, let me explain it in the context of Mendelian genetics. So if we were crossing white flowers with red flowers, and red is incomplete with respect to white, or has incomplete dominance, then the plant that is heterozygous, so a white allele and a red allele are present in the genotype, that plant would actually be pink. So it's intermediate between the red and the white. The heterozygous would be pink. In terms of physiological conditions, an example of that would be sickle cell disease, which we'll explain on the next slide. So in this case, the 2 forms of the gene are hemoglobin A and hemoglobin S. Hemoglobin A encodes for a normal hemoglobin protein, while hemoglobin S encodes for an altered and diseased form of a hemoglobin protein. So the hemoglobin A homozygous individual would be normal, have normal hemoglobin, normal oxygen carrying capacity. And the individual that is homozygous recessive for the S allele would have full-blown sickle cell disease, which is a debilitating condition with severe anemia, breakdown of red blood cells that clog arteries and cause multiple organ damage. However, the heterozygous individual that has 1 copy of the A allele and the other copy is the S allele, they would produce half normal and half abnormal hemoglobin, This individual is said to be a carrier for the disease, and they would have a mild form of sickle cell, so mild in some individuals that it doesn't even appear as sickle cell disease. It's undetected. So they have minor problems. They're said to be carriers for the disease because they carry one copy of the defective gene or the defective allele that they can then pass on to their offspring. And if we look at the Punnett square to the right, here we have crossing or mating between two heterozygous individuals or two sickle cell carriers. Note that they have a 1 in 4 chance with every pregnancy of producing a child that has full-blown sickle cell disease. They have a 1 in 2 chance, or 50%, of having a child that is a carrier as well, so they're heterozygous. And with every pregnancy, there's a 1 in 4 chance that that child will be normal with respect to the sickle cell trait. So both copies will be the healthy hemoglobin A allele. Another variation on the pattern of inheritance is multiple allele inheritance, where some genes have more than 2 alleles. So there's versions 1, 2, and 3, or in this case with blood typing, there's A, B, and O. The ABO blood type is also an example of codominance, where both genes are equally expressed in heterozygous. So of course the homozygous individuals will express whatever protein is encoded by that gene, but the individuals that have copies of the two different alleles will express both equally. So let's use the plant example one more time. If red is now codominant to white, the heterozygous plant, which has one copy of the red allele and the other copy is the white allele, will actually be a flower with red and white polka dots. In case of the blood typing, if an individual is heterozygous for the A antigen on the surface of the red blood cells as well as the B version on the other chromosome, then their red blood cells will have both A and B antigens or proteins on the surface. If the person is homozygous for the A, then their blood type is of course type A. If they're homozygous for the B, blood type is B. If they are heterozygous for A and B, as in the third box on our table, then their blood type is technically AB. Both antigens are expressed on the surface of the red blood cells. Now the third allele in this multiple inheritance pattern is the I allele, or the I gene. This form results in the production of no surface proteins or no surface antigens. So the individual that is heterozygous, for example, AI, will only express the A form. So they are also blood type A. Similarly, for the IB individual or the BI individual, they will have only be on the surface. But the person that is homozygous for the I allele will have neither antigen on their red blood cells, and their blood type is O. Okay, so again, A and B are codominant. They are equally expressed when they're present, and the I allele in this case results in no antigen on the surface of the red blood cells. Polygenic inheritance is very common. It's when inherited traits are controlled by more than one gene, so that several loci or maybe even different chromosomes, the combined effect of all of those genes plus the variance in the alleles of those genes will determine the final phenotype of the individual for that trait. This is also the case with complex inheritance, where you add to this mix environmental and nurturing factors in the expression of that gene. So most polygenic traits are also complex in that not only are multiple genes involved, but also environmental factors will affect the final expression of that trait. For example, skin color is not just due to the skin color gene. There are multiple genes that affect skin color, as well as hair color, height, metabolic rate, and body build. And you add to this things like diet, sleep during growth and development for height, the foods they eat, their physical activity. All of these things can affect these traits, especially height, metabolism, and body build. Even if a person, for example, inherits several genes for tallness, they can only reach their full height if they also have nutritional support and adequate sleep to support the activity of those genes. Other factors that can occur are nutrition, and in this case folic acid availability during early gestation. We mentioned that the embryonic period is very susceptible to defects and mutations, so inadequate folic acid will result in defects in the neural tube and neurological deficiencies in the offspring. So that's an environmental effect, even though the genes for normal nervous system development may have been present. This also is the source of a lot of variability in the offspring. So you can have children or siblings in a family that have a range of skin colors, a range of heights, a range of hair textures, etc. Because of that variability with the multiple genes that are involved, the polygenic nature, as well as the environmental factors. All right, let's finish up with looking at sex chromosome related conditions. So we mentioned that one of the 23 pairs account for the sexual features and the sexual identity of the offspring. So if the cells within the individual have 2 copies of the X chromosome, then they are technically female, and if they have one X but one Y, then that's a male. There are variations on this that are rare that occur with genetic abnormalities where they can inherit extra copies of either of these chromosomes, or sometimes are deficient in one. That will lead to males and females that are not fully developed in those characteristics. So when the gametes are formed in the male during spermatogenesis, the gamete can either carry an X chromosome or a Y chromosome. The father always produces sperm with either X or a Y, depending on how they segregate during meiosis. Females can only pass on X chromosomes. So that when the gametes unite, the zygote will always inherit an X chromosome from the mother, and then the sex of the zygote, or of the child that will be born, is determined by which type of sperm fertilized that oocyte from the mother. Is it an X sperm, then the offspring will be female. If it's a Y sperm, then it would be male. Also note that during fetal development or embryonic development, the embryos essentially look identical to about 7 weeks, and then they suspect that genes on the Y chromosome called the SRY are responsible for the male pattern of development, and the fact that the X chromosome does not have this region leads to the female pattern of development. Sex-linked inheritance involves traits that are encoded on the regions of the X chromosome that are missing on the Y chromosome because it's shorter. In this case, in the male that has an X and a Y chromosome, the genotype on the X chromosome will determine the phenotype because there's nothing on the Y chromosome to counteract it, to balance it, to mask it, for example, if it was a dominant trait. However, in the female, it is the combined effect of the 2 alleles on both X chromosomes that will determine the phenotype. So note that if the trait is X-linked and it's recessive, for example, color blindness, the male's genotype would be X color blindness, Y. That male would be colorblind. But in the female, if she was heterozygous for that trait and she had one normal vision X chromosome and the other chromosome carried the color blindness allele, that female would still have normal vision, okay, because the regular vision is dominant to the color blindness, but she would be a carrier for color blindness. That if she passed on that defective X chromosome with color blindness allele to her son, which would then be a son because it inherited a Y chromosome from the father, that son would be colorblind. So the mother had normal vision but was a carrier for color blindness. The trait only appeared in the son. Now in the next generation, that son could pass on the color blindness X chromosome to a daughter. That daughter, if it inherited a normal X chromosome from its mother, would be a new carrier. So the color blindness trait would not show up in that generation. And again, the boys in that generation could have normal vision because they are male when they inherit the normal Y chromosome from their father. So again, these sex-linked characteristics tend to only appear in the males, and they're passed from carrier mothers to the son. The son then usually passes on, does not pass on that trait to their children. It's always from mother to son, mother to son. Okay, I'll go through this more in class. And other X-linked characteristics, I have to look them up for you. We'll discuss them in class.